we have a presentation from Aardvark Therapeutics from Dr. Tien.
Hi, great to be here. Thank you so much. Let's see, advance the slide. All right, so Aardvark, San Diego-based biotech company, and we're developing ARD-101. It's a oral, twice daily bitter taste receptor agonist. It's a very unique drug with some special properties. It's a drug that's 99% gut restricted, so you take it orally, but 99% of the drug is retained in the gut and eliminated with bowel movement, but it doesn't work despite the fact that it's gut restricted. It works because it's gut restricted, because it's invoking gut-brain signaling via gut-brain CCK, and we think that's actually especially important for Prader-Willi syndrome, which I'll go over in the later slides. Currently, we're about to embark on the pivotal phase 3 study, which will be a multinational study, hopefully targeting to complete by first quarter of 2026. And the central theme of our company is that there is a difference between appetite and hunger. Generally speaking, folks won't really delineate the difference between appetite and hunger, thinking that there may be just different extremes along the same spectrum, but we believe that there are different neurologic drives that govern behavior. So hyperphagia could be driven by either appetite or hunger. Generally speaking, appetite is your body chasing pleasure, the carrot, and then hunger is your brain running away from a painful stimuli. Appetite, the way to kind of illustrate that fact is if you like chocolate cake, that appetite's driving you to eat that chocolate cake. If that chocolate cake were to turn into dog food, I don't think anybody would go for that. So if worldwide all the food turned into dog food, there'd be very little obesity in the world because people wouldn't be compelled to eat too much of that. But if you were trapped in a room with only dog food to eat, eventually hunger would compel you to eat that. And we believe that a lot of the GLP-1 programs are actually diminishing appetite. There are lots of lines of reasoning for that. You see ghrelin actually paradoxically increasing over time as a compensatory response to the anti-appetite effect of the GLP-1s. It's also decreasing the appeal of food. You see increased nausea, and then self-reported appetite goes down, but it's differentiated from hunger. Whereas hunger is governed by CCK, gut-brain CCK invokes satiety in a different manner, so it basically takes away the penalty of not eating. And for a variety of reasons, which I'll talk about in the next slide, we think in Prader-Willi syndrome, hunger is the dominant driver of the hyperphagia, and it's actually related to the mechanism of how our drug is working. So actually worked by Tony Holland 32 years ago, looked at CCK, and as he mentioned in his opening slide, they have maybe a basal level of CCK that's similar to normal individuals, but with food, the response is diminished as compared to a normal individual. So when they eat food, you don't have the proportional rise in CCK to normally invoke the satiety, which is why they have this perpetual unabating hunger. And we believe invocation of a secondary switch to induce endogenous release of CCK is able to help ameliorate the hunger that the Prader-Willi syndrome individuals experience. And there's actually more lines of evidence that kind of suggest that this is the case. If you knock out CCK receptors in rat models, the rats will actually have unabating eating behavior as well too. And not only does CCK, gut-brain CCK, regulate hunger, it also has a lot of other functions in your body, including induction of gut motility, gallbladder contraction, maintenance of lean body mass, and even some anti-anxiety inference in terms of the role of gut-brain CCK. And as we know, Prader-Willi individuals have slow gut motility. They get gallstones at an early age, even at a certain body mass index, they'll actually have proportionally greater body fat percentage, and we think that's correlated with a deficiency in afferent CCK signaling. So our drug is working through gut-brain signaling, so we're targeting bitter taste receptors. So more on bitter taste receptors, it's a very evolutionarily conserved GPCR. All mammals have bitter taste receptors. There's 25 different subtypes, and generally they invoke a appropriate response to toxins in the environment. So they're actually located throughout your body, not just your oral cavity, and it's an adaptive response to toxicity. It's actually more difficult than one might imagine to find a very potent bitter agonist that's also safe, but that's what ARD-101 is. So your body is treating it like it's a toxin, but it's not, and we don't want patients to taste it. So it's masked so that you're not aware of the bitterness when you ingest it, but it's hitting the gut bitter taste receptors to invoke this signaling. And the receptors for bitter taste are on the lumen of the gut, and it is targeting enteroendocrine I and L cells and invoking a basolateral side local secretion of CCK, which in turn hits vagal afferents, which project to the nucleus of the solitary tract to shut down hunger. And this is very important because of the selective anatomic expression of CCK, because CCK has been tried as a therapeutic intervention before, but the problem with CCK as a systemically administered analog, unlike GLP-1, is that you have a lot of on-target, off-tissue effects that will not be tolerated. So if you have a systemic CCK agent, and this has been tried with various companies over the last 30 years, you have hallucinations, neurotoxicity, pancreatitis, and this is related to the unintentional effects of a systemic CCK. But since we're anatomically hitting CCK only in the peri-gut region, we're anatomically restricting where that CCK is secreted. We've now had clinical experience with a variety of patients, including normal healthy volunteers, Prader-Willi individuals, as well as obesity and post-bariatric surgery patients, over 70 patients so far. No serious adverse events, nothing higher than a grade 2. We only had 3 grade 2s, which were itching resolved with Benadryl. So when you intervene, it's automatically a grade 2. Other than that, we think the calculated therapeutic index of ARD-101 is approximately 80, which makes it technically mathematically safer than a lot of common over-the-counter medications. And we think that that's actually one of the strong suits of the drug is because of its safety. And that safety is contributed because of the fact it's gut restricted. So you don't have to worry about renal tox, in theory, liver metabolism. There's fewer drug-drug interaction worries because it doesn't really escape the gut as far as we understand. And that's why we think it's, and also it's not mutually exclusive with other approaches because mechanistically it's differentiated and orthogonal. So we do think that it is additive to other therapies and potentially synergistic with some others as well, given how it's working. And also in the natural set of gut peptide hormone release, it's generally a concomitant secretion with food ingestion along with GLP-1, and there's really not an intervention that can successfully invoke endogenous CCK release as far as we know. Of the clinical experience we had, our first study in Prader-Willi syndrome was in 12 patients at 2 different clinical sites, one at Stanford with Dan Stafford and Sean McCandless in Children's Hospital in Colorado. And of the 12 patients treated, this was an open label single arm study at the low dose, which is 200 milligrams BID. And I want to emphasize that fact because we now believe that 800 milligrams BID is a much more effective dose. We have a lot of data showing us that we should have expectation that a higher dose should do better. And given the calculated therapeutic index, we still think that's a very safe dose. So high dose is still low dose, but it's higher relative to the low dose. But even with the low dose, 11 out of the 12 patients had a reduced HQ-CT and average reduction of 7.1 in just a 28-day period, because when we actually did that study, we had only had the safety database for 28-day dosing. Obviously, we're going to intend to dose much longer. We're running the 9-month GLP tox studies now, which will eventually allow us, hopefully, to give indefinite dosing. But we're looking for a 3-month efficacy endpoint for our pivotal trial. And of the 12 patients, as I mentioned, 11 had improvements in their HQ-CT, and 4 of those patients had almost near complete resolution of their hyperphagia and a very remarkable responses to therapy. And anecdotally, even though we only looked at the patient response at day 14 and 28, it seemed that for the patients that saw the effect, it was observable very early on in treatment. Even the first day of treatment. And we also saw trends of reduced body fat percentage even in that 28-day period, which is consistent with what we think gut-brain CCK should do. And because we think we're restoring normal gut-brain CCK tone, we also saw, even though there wasn't a declared secondary endpoint, we saw anecdotes of decreased anxiousness, reduced inflammation, and we didn't measure gut transit time, although we think mechanistically it would be very rational for us to see improvements there as well. And also, I forgot to mention, we have a 6-patient extension study now looking at dose escalation up to 800 milligrams, so we should have that data shortly. But our pivotal study is not contingent upon the completion of that study, but we are dosing the last patients in that study for the expansion trial for that. And just also want to say that our clinical experience in other patient populations recapitulate our expectation that we are decreasing hunger versus appetite. This is data from our randomized placebo-controlled trial in general obesity patients. And these patients, lost a modest amount of weight, like about 1% in a 28-day period. There was no diet and exercise in that protocol, but patients had reported a statistically significant reduction in self-reported hunger. This is just general obesity patients, not Prader-Willi, and that was observed versus placebo. And ghrelin levels, which we think are reflective of hunger state. There's generally proportionality of ghrelin in terms of perceived hunger. As we know, Prader-Willi individuals have elevated ghrelin levels. We saw reduced ghrelin versus the placebo group measured 2 different ways. On the left, you'll see patients were fasted overnight and then they were skipping breakfast. And as you would expect, the placebo patients had increased ghrelin, whereas the ones treated with ARD 101 had their level flat, and that achieved statistical significance. And also comparing day 28 to screening ghrelin, we also saw reductions in the ARD 101 treated group, even though those patients had lost 1% of their weight relative to the placebo patients. Again, ghrelin is going the correct way, we believe, if it's reflective of decreasing hunger. And this is some of the evidence why we think that, that we see dose response. So higher doses do better in preclinical models. We think humans will be the same. And again, we're off to the races with our pivotal study, and we hope to enroll very soon. That's it.